Okay, next up, we have Jill Gallaher from Moffitt, who's going to tell us about selection of evolvability as a trait in cancer growth and treatment. It was, yeah. For you, maybe.
There we go. Thank you. Yeah, so this is kind of interesting topic that I've kind of come back to after a while. A lot of my modeling that I've been doing lately is rooted in at least some data or trying to understand some biological mechanisms. While this is kind of, in light of maybe the cartoon that Jake put up, I like to think of it now as just a higher purity of model. But, unclouded by data, but actually thinking about some very deep questions that maybe we can't get to data yet, but thinking about these ideas and how it relates to tumor growth and treatment. And that's evolvability. And so there's several definitions out there that I found that have slightly different definitions. In 2010, it says it's concerned with the selection of new phenotypes, requires the ability to generate distinct phenotypes, and requires that some of these have a non-negligible probability of being selected by the environment. And another one, they say it's the ability of a biological system to produce phenotypic variation that's both heritable and adaptive. And this last one that I really like kind of got me thinking about, what to, how to implement this in the tools that I have is that it represents a trait by which the generation of heritable phenotypic variation itself becomes heritable and subject to natural selection. So evolvability, not just being phenotypic variation, but being a trait that itself can evolve. And so the tool that I'm going to use here is just this simple off-lattice agent-based model. Cells basically go through this diagram at the top, and if there's space for them to divide, once they are mature, once they hit their clock, they will divide unless there's a drug and they're sensitive to that drug, and then they might die. If there is no space, then they'll just go into this quiescent state until there is space. So pretty simple stuff. So just taking that model and growing monoclonal populations of different combinations of proliferation and migration. So I wanted to look at this proliferation migration space because it's kind of the most fundamental traits that you might have in a solid tumor that matter is how fast is it proliferating and how fast are you moving out. If you grow these different combinations of monoclonal populations to the same time point, then you end up with these this kind of surface of a fitness landscape. And you could say fitness is like the growth rate, and you could look at these four extremes, and you see the one at the top is really big, the one at the bottom is quite small, and these two on the sides they're actually the same. They have the same growth rate, but in different ways. And so if you look at the plot on the right, I'm here showing the proliferating fraction. And so you can see these green versus yellow cells, and there's a lot more green on the one to the top right. And so that's just showing that there's two different ways to get to the same growth rate. You can have a lot of cells proliferating at a slower rate, or less cells proliferating at a faster rate. But you can't always get to that whole fitness landscape. It's not always available. And so we talked earlier about trade-offs, and if they exist, sometimes they do, sometimes they don't. It kind of depends on the context. But let's say that there are these certain areas that are cut off and not allowed by the certain cancer cell or bacteria cell, whatever we might be talking about. If not, then you start to shape the space into different regions that makes a difference in the evolution and the trajectory of these populations. There could be, more generalists that are found if it's still allowing that top corner to be attained, or you could get specialists that are better at proliferating, not so good at migrating, or better at migrating and not good at proliferating. And if you look at the bottom here, I'm also comparing what happens when you increase the death rate. So if you do have this continuous improvement and you can get to that top peak at the fitness landscape, then you're always going to find that peak. These cells will be the most fit. But as you start to cut off that space, some of these small variations in that landscape really matters. And so because you have this high death rate, you select for more proliferative cells. And when you have only these specialists that are allowed, then that high death rate will select for faster proliferators rather than faster migrators. And there's other things that affect this landscape. So the environment. So let's say you have a drug that targets fast proliferators, a chemo, or you have a drug that targets fast migrators. In this model, if you just have these monoclonal populations and you subject them to these drugs, then you get landscapes that look like this. So now the corner that you would be seeing on the previous, right, I've kind of flipped it around. So now you have basically, a different landscape that shows how these different populations are fit in these different drugs. That little yellow line kind of shows where the growth is 0. And if you're above that, then you'll have a positive net growth rate. Otherwise, you're going to die. And basically the root of all of this is trying to think about how you would use this eventually to strategize about treatments, and that maybe you consider different environments and different landscapes when you treat, and that when the tumor is growing, you notice that it's going to go up this peak. As you start to apply an anti-proliferative drug, it might move this way, and an anti-migratory drug, it might move up the peak in that way. But there's something wrong about this whole thing that I'm showing you, is because these are all monoclonal populations. We know that tumors are not monoclonal. There's heterogeneity, there's phenotypic heterogeneity. So we can't really capture that in a fitness landscape like this, but imagine that, what I've shown you, there's really no movement on here because they're only monoclonal populations. But with heterogeneity, there is movement because there's selection, and there's spatial differences, especially when we're looking at migration and proliferation. So that's kind of what I wanted to consider here, and the movement through this space. Oh, and in avoiding that little area right there that is completely untargetable, that's always above a positive growth rate, it never gets negative. So I'll get to my definition here using this tool of evolvability. And in this case, the way that I would say is it's the way that a daughter cells will inherit the parent trait. How far can you fall from the tree? If you have direct inheritance, you're going to inherit exactly what your parent had given you. Otherwise, there's a probability that you could inherit a trait that's a certain distance from where you are. And just to preempt for when I show you some trade-off stuff, if you fall over the edge, you just roll again. This is just a random sampling. So that's the assumptions that I'm making there. So what I'm showing you here is 3 situations with evolvability at increasing towards the right. So here I'm starting them all in this little lower corner with low migration and low proliferation. I'm just going to start that again because it's really fast. Or am I? Oh, it wants to finish first, really? Okay. All right, so you can see as you can have these like larger jumps, this higher evolvability that, you always stay in the corner. If I have 0 evolvability, as you kind of increase it, you can have these different phenotypes that that get selected for subsequently. And then on the very right, you see all this diversity just because there's so much jumping all over the space. And so it can get really high. What's the consequence of this? Does this exist in reality? Perhaps. But just looking at it mathematically and how it moves through that space is kind of what I wanted to. I don't know why I tried to fix this, but you've already seen it because I just showed you it on the previous slide. So when you have an open trade-off, so no trade-off, basically, this is what happens. It moves towards that high proliferation, high migration. And the other two, I'm showing you different trade-offs. So with the convex trade-off, you can actually see it split into these two different, some that are faster at migrating, some that are faster at proliferating. Once I do this again, I'll grow them larger, but this is what we have right now. For the concave trade-off, you see that at least for this set of parameters that I've chosen, it moves towards this fast migration. So space is a limitation. And so being migratory is really important in this case. And as I showed you before, if I were to increase that death rate, it would move towards that faster proliferation. But looking at this concave a little bit more, because it's interesting, is that if we look at then the movement through this phenotype space, so how fast you can move through the evolvability, if we're increasing towards the right, I did press it, let's see. Right, so on the right, you see it moves towards that high migration. As you start to increase evolvability, it actually moves towards the faster proliferation. And if you go further, there's so much jumping around that it doesn't really matter. And you kind of get all this heterogeneity and it stays, it kind of has the most heterogeneity throughout that space because it doesn't have to remember. Part of moving through this space that can be both beneficial and detrimental to a tumor is remembering where you're from, where you came from prior. So now I take these tumors and apply an anti-proliferative treatment. So what you see here, faster evolvability. So this, really far jumping from parental traits on the right. And you see that the one on the left basically is selecting for these less fit cells, but then kind of moving up towards these migratory cells. What's interesting here on the right is that eventually you do have kind of this coexistence between fast proliferators and fast migrators. I think it's something similar to like what David was showing, where you have a little protection. So these cells kind of move out and provide a little refuge for proliferators to move in, and it grows that way. And then the last thing I wanted to look at is evolvability as a trait. And what I've shown you so far is evolvability where it's allowed to go up and down. And so that's what I'll show you on the top. On the bottom, it's only continuous improvement. So it only allows to go up. And I guess I need to hurry. So at the bottom, you can see that the only allowing it to go up in evolvability basically selects for a really heterogeneous tumor. It makes it grow pretty fast. While the one on the right, the evolvability that's selected is actually not the fastest evolvability. Once you get up to that top fast proliferation and fast migration, you don't have anywhere else to go. It doesn't help you to move away from that.
What's happening there? There's a third axis that's also.
Say what?
There's a third axis.
Oh, yeah. So it's proliferation and migration on the left that I'm showing you. On the very right, it's evolvability. So as it goes from white to blue to red, it goes to higher evolvability. So basically that cell inherits that evolvability, which means that if your parent can jump really far from where you were, so can you. And so, yeah, that's the difference, is that at the top, it doesn't really, it kind of finds a jump that's optimal versus the one on the bottom where it can only go up, it can only increase in its jumps. So then what happens next when you apply treatment to these two tumors is, if this is if I would have done it again, it would have been a longer run simulation. But anyway, on the one at the top, it does what you would think would do is it selects for this slow migration, slow proliferation, and kind of comes back. What's happening at the bottom and why I should have run it longer is because it takes so long, but it is slowly declining in population. So the problem here on the bottom, having this high evolvability, is that it doesn't move into a region where it can survive. It keeps jumping into the region where it dies. So it's not learning, it doesn't have that memory to recognize that it shouldn't keep going there. So having this higher evolvability is no longer beneficial. And so with that, I just wanted to thank Sandy and my colleagues, Max C, who's now at Cleveland Clinic, and Jeff and Mark for all the discussions, and that's it.
Great job, Joe. I'm curious about the sort of almost like it's melting down at too high an evolvability. Can you kind of comment on, I know that it's a complex model, but do you think a simpler model could allow you to sort of work out what that threshold would be where evolvability starts to hurt you?
It's not so complicated. But I mean, there's a lot, yeah, there are levers that you can keep pushing, you know, when you start to think about death rates. And but I do think that, yeah, trying to find where that is, I think is really important, because, you know, originally I wasn't even thinking about evolvability. I was just thinking about treatment sequencing when I saw this happen at first, and like inheritance. So just thinking about resampling versus, you know, being able to drift through the space. And so as you kind of just have like a mean and you resample from the trait, like the tumors would always die. And it seemed like, well, why? It's just because they're just jumping into a space where they're gonna die. But it doesn't always happen. So yeah, there are spots, and yeah, that's something I'd like to find, is where it matters, and yeah, where it fails or where it succeeds in that. Yeah. Thanks.
I have a different question, but maybe as a follow-up to that. So that should be very similar to genetic meltdown, right? Yeah. And they're very interesting spatial effects with genetic meltdown. That's okay. So
yeah, it's interesting because I have thought about putting that in there specifically, is having like too fast of an evolvability would actually increase the random death rate, because it makes sense. There's some biological backing for that.
So maybe lunch conversation.
Oh, sure. You had a different question.
So yeah, my actual question was, so you have probably just a very linear dispersion kernel, right, for the migration, but also in moving through phase space. So I think you had like a plot where it's micrometers per hour, something like that. Migration, yeah. Right. So how would that change if you have different kernels? So if you have like, you know, with a very long tail, what people have looked at for, you know, virus dispersal, etc. So would that then change the dynamics at all?
So you mean how so I choose to jump through here at regular intervals? Yeah, is that what you mean? Like in proliferation?
Kind of, I was assuming that's what you did. But,
yes, it is. Yeah.
Okay. Okay.
Yeah, so that's really interesting. And, you know, when I first started working on something like this, we were not thinking about proliferation as like a doubling time, but as a intermitotic, well, as more of like, well, a proliferation rate. And so as you kind of shift from a proliferation rate to a time, it does like adjust the way like that a lot of cell distributions are found. I don't know. It's an interesting question I hadn't thought about. I was just doing the most simplest thing. But but especially when you start to combine two things, the migration and proliferation, yeah, how it matters how you jump through the space.
Because I think it actually might help with, you know, with kind of what Jake was asking, because then you can explore more space, extreme space, to still go somewhere, but the average draft that you have is still contained so that you don't have any meltdown, right? So that might be a good strategy to evolve towards a long-tailed dispersion kernel, actually.
Oh, interesting. Okay, yeah, I'd like to talk more about that. Thank you.
That was fantastic visuals. Thanks.
Oh, thanks.
Still really great work. I guess sort of two comments. One is I really like the version where the evolvability can go up and down, because I think that's realistic. We know in biology that you can both increase and decrease mutation rates, for example. But in particular, I think your results replicate something we've been seeing hints about with respect to genomic instability. That is that we think we certainly see an increase in genomic instability during progression, but we think that when they're getting, the cancers may find some fitness peak, and then there'd be selection to reduce that instability. And I think Nick Davin's single cell data suggested that even though these clones are highly messed up, they seem to have evolved a stable messed up karyotype, where they had to sort of stabilize their genome. So I think we see in cancer what you saw there, that as it climbs the fitness peak, is a selection for higher evolvability, but then when it gets to the top, it gets selection for lower evolvability.
Oh, that's really interesting. And I wonder too how there's progression and then there's metastases and finding a new niche and how it matters through there.
Yeah, every time you're just sort of switching your context, should see selection for a higher evolvability until you optimize for that new context.
Yeah, that sounds great. Thanks.
Hi, thank you for your talk. One of your slides talking about the more conceptual motivation mentioned this question of, can we steer phenotypic behavior by changing environments? And I was curious if part of this project took a look at like a stochastic optimal control framework for answering that question. Like I'm thinking there's a couple of papers I've read looking at that question of kind of steering phenotype, but in the context of immune system and pathogen recognition, but it's kind of a general mathematical framework that can be applied to something like this. So I was just curious if there is like an optimal control facet.
I hadn't thought about it in this context, but yeah, I don't know. Yeah, let's talk about it.
Yeah,
Thanks.
just
Yeah.
curious. So cool.
Thanks.
Thanks, Jill.